In this video, we'll review the stages of the cell cycle and focus mainly on recognizing the different stages of mitosis. The first stage of the cell cycle is interphase. During interphase, DNA is in the form of chromatin. The cell spends most of its life cycle in interphase. The cell is performing its normal everyday functions and it's preparing to divide. Some of the changes that take place in the cell while it's in interphase and its preparation to divide is that it grows in size. Another very important step that takes place is that DNA replicates. This ensures that the two new cells formed after cell division each have their own copy of DNA. The next stage of the cell cycle after interphase is mitosis. Mitosis, by definition, is the division of the nucleus. It has four substages: prophase, metaphase, anaphase, and telophase. In this video, we're going to focus on recognizing each of those phases. The last stage of the cell cycle is called cytokinesis. Cytokinesis is when the cytoplasm is pinched in half and the end result is two daughter cells. Cytokinesis is really difficult to identify. Most times it's grouped in with telophase because it's simply a very late telophase when the cytoplasm is pinched in half. Here we see a cell in prophase, the first phase of mitosis. A key characteristic of prophase is that chromosomes are visible. Remember that during interphase, DNA is in the form of chromatin, a stringy, fibrous, thread-like form. In order for the nucleus to divide properly and separate the replicated DNA easily, the DNA condenses and coils around proteins, forming rods. These rods are chromosomes. We often see chromosomes drawn as X's in diagrams. The X structure signifies two identical parts to each chromosome. These are called sister chromatids. We have identical sister chromatids because of the replication process that took place during interphase. As the cell continues through mitosis, we will eventually see the sister chromatids separate. Other changes that take place during prophase are the formation of spindle fibers. Spindle fibers are modified microtubules formed by centrioles. We see that the centrioles have replicated and the spindle fibers form between them. The nuclear envelope breaks down, allowing the fibers to attach to the chromosomes. The photo to the right shows an actual cell in prophase. Notice that chromosomes are visible and no nuclear envelope can be seen. We do not see the centrioles, however, because of their small size. Near the end of prophase, the spindle fibers have all attached to each chromosome and the centrioles are moving to opposite sides of the cell. Metaphase begins with this movement lining the chromosomes up down the center of the cell. Metaphase is fairly easy to recognize because the chromosomes appear as a dark line down the center of the cell. Anaphase begins with the contraction of the spindle fibers. Since the spindle fibers are attached to each replicated chromosome, the shortening of the spindle fibers separates them. The cell elongates and we begin to see a lightened area down the middle of the cell with a set of chromosomes on each side. Telophase is the last phase of mitosis. Once the chromosomes are separated, a new nuclear envelope reforms around each set. We now have one cell with two nuclei, and the cell is ready to separate into two cells. A cleavage furrow forms along the center of the cell. The cleavage furrow is this indentation seen at the top and bottom of the cell. When identifying telophase, look for the cleavage furrow. It's the easiest characteristic to see. Following telophase is cytokinesis, which is the actual splitting of the cell. Oftentimes, telophase and cytokinesis are grouped together because it's simply the deepening of the cleavage furrow that eventually separates the cell. In this image, you're seeing many cells, each within different phases of the cell cycle. Hopefully, you're already able to pick out a few of the mitosis phases. This cell is in metaphase. Notice the dark line down the center of the cell. This cell is in anaphase. Notice the lightened space down the center with two sets of chromosomes separating. This cell is in prophase. Notice the dark chromosomes seen in the center of the cell. Most all of the other cells are in interphase. If you remember, interphase is the first stage of the cell cycle. It's also the longest stage of the cell cycle. This is when the cell is performing its normal functions, DNA is replicating, and the cell is preparing to divide. Since interphase is the longest stage of the cell cycle, it makes sense that most all other cells would be found in interphase. To recognize interphase, you're simply looking for a cell with a nucleus on the inside and an visible nuclear envelope. This cell is in interphase. This one's in interphase, this one's in interphase, this one's in interphase, this one is, and this one is. You'll notice almost all of the cells are in interphase, with just a few of the mitotic phases scattered in. Here we see another slide of many cells. Notice again that most of the cells are in interphase. There are, however, a few phases of mitosis found in some of these cells. This cell is clearly a telophase. Notice the cleavage furrow here at the bottom and here also at the top. This cell down here in the corner is a prophase. Notice you can see chromosomes found within the cell. Are you able to pick out a few phases of mitosis on this slide? Hopefully you see metaphase here, here, and here. And most of the surrounding cells are in interphase like the others. There's also a prophase present right here with chromosomes on the inside.